This Week in Virology, the podcast about viruses, the kind that make you sick. From the Third International Symposium on Infectious Diseases in Bats in Fort Collins, Colorado, this is TWiV, This Week in Virology, episode 923, recorded on July 26th, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today here in Fort Collins, Brienne Barker.
Hi, Vincent. Great to see you. It is a lovely day outside, bright and sunny, 84 Fahrenheit, 29 Celsius. But lucky for us, we get to have a great conversation inside.
It's lovely weather here. Thank you, Tony, for the lovely weather. We really appreciate it. So we have two guests for you today. This is, as I said, a meeting about infectious diseases of bats and actually much more. And we're going to talk about that. And so from the NIH in Hamilton, Montana, Vincent Munster, welcome.
Glad to be here, and thanks for inviting me here on stage.
Welcome back. Actually, you were on TWiV a long time ago. February 2017.
Yeah, a lot has happened in the meantime.
Oh, yes. Oh, yes.
Couple things.
And Vincent is the only other Vincent who's ever been on TWiV, I think. And now I know I'm going to be proven wrong about that. But you think it's okay, right, Brienne?
I think it's okay.
Yeah, I'm the one with the weird accent.
Also joining us from Cornell University, Raina Plowright. Welcome.
Thank you. It's great to be here. And I'm the other one with a weird accent.
Well, it just goes to show that research is international, right? Which is as it should be. Everybody in the world should be involved. So let's talk about bats, bat viruses, and spillovers. Let's start by histories. We love to hear how our guest scientists have been trained and they ended up at this point. Raina, tell us about your training.
Well, I trained as a veterinarian, and then I decided to do a graduate degree in epidemiology at the University of California, Davis, which is what brought me to the United States. And while I was doing my graduate studies, I thought I'd better learn something about ecology, since I was studying wildlife and their diseases. And that was it. Ecology it blew my mind to know that there was viruses were part of these complex systems, everything moving in space and time and interacting with environment. And so I actually changed my PhD to ecology, finished epidemiology as a master's, and since then I've been a disease ecologist.
Did you always know you wanted to do science?
Yes, I was one of those people who wanted to be a veterinarian since I was like 6 years old, and I then made my way into science from veterinary medicine.
Got it.
What was your first academic appointment?
At Montana State University.
So you were there many years?
Oh, about 7 years.
Okay.
Yes, I've only just moved to Cornell University.
Very good. I know Cornell moderately well, having graduated, but many years ago. 1970, when did I graduate? 74, and many of you in the audience were not born yet, probably. Yeah. Vincent, how about you?
Well, I was trained as a molecular biologist and actually started working on epigenetic phase variation of outer membrane molecules of E. coli. So I think it's pretty far removed from what I'm doing right now. And then I ended up in a PhD student position at Erasmus Medical Center, working with Professor Ron Fougier and Ab Osterhaus, working on avian influenza. And I started with my training as a molecular biologist, very molecular, looking at HA and NA. But then there were all these questions. At that time, we had the first introduction of highly pathogenic avian influenza in Hong Kong. It started getting a bigger problem, and we were actually kind of surprised how little information there was where these viruses came from. The Netherlands is actually really strong on the ecology level on migratory birds. So I actually started contacting bird ecologists, and that's where, for me, sparked my interest in blending molecular virology classical virology, so really studying the virus in animal models, and then doing field studies, so going out into the field, making like comparison, for instance, what's happening in the backyard in the Netherlands, which has a lot of wild waterfowl, for instance, what's going on in the high Arctic. And then I wanted to do a postdoc, and I want to kind of like conceptually take that with me, but do something else. So I started then moving to Rocky Mountain Labs, the NIAID lab, which is a maximum containment lab, so that's a biosafety level 4 lab, and started working in the lab of Heinz Feldman. And I thought, well, bats fly, so I guess there's at least some kind of relationship with birds. And I never really looked back.
And how did you get involved in science? Did you always know you wanted to do it?
No, I was actually like, as I guess like most guys, I was a little bit slow. And then I kind of figured out, hey, I actually like doing science. I like doing it a lot. And I think I'm actually reasonably good at it. But I didn't really have like a really career trajectory in mind. I was just going from step to step, and I kind of rolled on until here I am for the second time in TWiV.
And Ron Fouché was also on TWiV several times. So we keep it in the family sometimes, right? All right, so I want our listeners to come away with an appreciation for bats and all that they do, because as Peter Daszak told us, it's not always the case, at least from the journalism, you don't get that. So let's start with a bunch of questions about bats and bat viruses, and then we'll move into ecology and spillovers, okay? So first of all, and you know, the two of you are on many papers together. It's very interesting. And so we may ask you both to answer the same question. So first, Raina, how are bats beneficial?
Bats are really important for our ecosystems and therefore for us. So bats are pollinators and seed dispersers and insect consumers. In fact, some of the systems that we work in, they're the only creatures that can move the genetic material of plants across huge distances across fragmented ecosystems. So they're really important in terms of climate change and being able to develop the resilience of forests to climate change. Bats are going to be absolutely critical.
So what would happen if you removed all the bats from the planet?
It would be absolute disaster. We would lose those pollination services. Many plants would lose the ability to reproduce. We would have insect problems. Many of these insects are also problems for agriculture. So that wouldn't be a good idea at all.
How long do bats live?
Well, bats are really unusual in that they're very small-bodied but have very long lifespans. So usually, the bigger an animal is, the longer its life is. Like an elephant, lives 70 years or so, and a mouse, a couple of years. But bats actually they're very small, but can live, like the Brand's bat lives up to 41 years. So that's very unusual. And the other outlier is the naked mole rat.
That's like 30 years, right? Something like that. I heard about that recently. The naked mole rat.
I don't know what the lifespan of a naked mole rat is.
I think it's something like 30 years.
I assume we'll get letters.
Yeah. Okay, Vincent, here's the $1,000 question. Do bats have a greater diversity of viruses than other mammals?
I think I would be, it's a little bit hand-waving here, but I think most data up to now would suggest yes, but I think what we're really missing is like really carefully designed studies to actually address that question, and then, for instance, do like a comparison, for instance, between rodents who have like a slightly comparable diversity of species versus bats. I think the bats will probably come out on top because they just have like completely different population structure, completely different lifestyle and contact rates, which potentially drive the diversity in these bats. But up for now, I'm still thinking that careful designed studies to actually address that specific questions are still needed.
And it might not be the diversity so much too as how important those viruses are for us, right? I think Kara Brook just presented a paper showing that they're more likely to host viruses that are very pathogenic in humans and maybe more likely to cause pandemics.
So why is that?
That's a good question. So we're going from $1 million question to the next?
Exactly, yeah.
Yeah, I don't know. I think like I think it's easy then to come up with a follow-up. If you really look at the diversity, which we're really targeting, looking very carefully in these bats, and we see this huge diversity, probably such an infinite small number of these viruses are pathogenic for humans. We're very good in detecting pathogenic viruses in humans once they spill over, because that's when we see them. So we see the Ebola outbreak, we see the Marburg outbreak. But then making that same inference of viruses who look very similar, let's say in the example of coronavirus, we have TWiV1, we had SAHL14, they have all the hallmarks of potentially being pandemic, but if you carefully study them, they still fail to come up to that level. And I think that's really, and that's of course why we are here all together in this research conference. I think that's still the big missing step in our ability to predict what's going on. Those small steps which like really separates a zoonotic or a cross-species virus, so something which spills over and infect 1 or 2 persons, for something which actually has pandemic or epidemic potential. And we still don't clearly understand what that difference is.
Just to follow up on the rodents, though, I think they may be equally important, right? Many zoonotic diseases come from rodents, and I think we need a infectious diseases symposium on rodents, right?
Yeah, which there are, and I think there are actually several people in the room. I saw Brian, a man in the back, do a lot of hantavirus studies. Obviously, we now have monkeypox emerging. Typically, as a virologist, we're kind of host reservoir agnostic. So I also work on rodent viruses, bat viruses, and obviously with Middle East respiratory syndrome started working on dromedary camels. So size-wise, we cover everything, I guess.
Yeah. All right. As everyone knows, bats are hosts for 3 of the 10 groups of viruses designated by the WHO as of pandemic concern. We have Henipaviruses, filoviruses, coronaviruses. So, Raina, are these viruses or related members in all bats all over the world?
No, actually that's very patchy in both space and in time. So why don't we see Ebola outbreaks in the United States? Well, there aren't the right hosts and there aren't the right viruses. So we don't know a lot about the location of many of the Henipaviruses, for example. We know that they're spread in bats across the Old World tropics, but we haven't characterized many of those viruses. So we could have zoonotic viruses in places that we're not aware yet. But in general, it's very hard to find these viruses in nature.
So I guess a follow-up would be, have we done enough surveillance to know the extent of, there are patchwork, as you say, but can we be confident given our extent of surveillance?
We've hardly done any surveillance for these viruses. So no, there's a massive diversity of viruses out in nature, and we have very little understanding of the capacity of these viruses to infect humans or spread amongst humans.
How has the COVID-19 pandemic impacted that?
Well, I don't know so far. I don't see a whole lot of money coming in to do more comprehensive surveillance. So I'm sure that there is going to be more investment, but I haven't seen something at the extent that I would have expected.
Vincent, here's another $1 million question. Do bats really tolerate all virus infections without getting sick?
Well, I think we saw some really nice data from RKI today, which suggests that there is still a little bit of an impact. I think that's, again, designing very careful lab studies, but we're actually not that good in determining whether a wild reservoir host gets sick or not sick. And one of the studies we did with avian influenza, and we were looking with these wild bird ecologists, if you look at an infected, in this case it was a Buick swan, you look at that swan, beautiful white swan, you don't see anything. It's infected, but it looks perfectly healthy. Then we put a satellite telemetry collar on them. We could actually see that the non-infected groupmates of that swan moved around from where it was way earlier, weeks earlier than the one which was infected, suggesting that yes, they might on the outside look very like asymptomatic, not sick, but could actually have a distinct ecological impact. Because typically with these animals, the earlier you get to your breeding grounds, the higher the success rate of your offspring is. So it could actually have like quite big impacts on the ability to generate offspring. And these are the things we're still not very good at looking at in bats. Other than obviously like the studies in Uganda, I think is a really nice example how we can move those kind of questions in the field. That's John Towner's studies with Marburg. And I think these are really nice framework which can like point people in the direction how to do these studies and not just do surveillance, but really like collect like life history data, potential distribution data, migratory data depending on the bat species. So I think there's still a world to discover.
So it's basically going to be a combination of field work to observe bats and also laboratory work where you challenge bats with viruses. And we heard today you can infect bats with Ebola virus and they live, which is stunning, frankly.
Yeah, but you can also infect mice with Ebola virus and they live as well.
Yeah.
Right, so that's like go into the literature and then you actually see parallels. So you see parallels, and why is that? Obviously, we don't know enough on the interaction on the molecular level, and that's of course where all these like lab studies come in to really tease that out. What's going on in the innate mechanism? How do the specific host, because like we're talking about 1,400 different potential reservoir species, which all respond in either slightly or vastly different ways to a different virus insult.
So Raina, I really enjoyed your talk yesterday, and I know we're going to talk a lot more about spillover in a bit, but on a simple level, how do bats actually shed viruses, and how do people get infected?
Well, the viruses are excreted through the things that come out of bats. So, for example, we work on the Hendra and Nipah viruses. They're excreted in the urine of bats. The coronaviruses are excreted in the feces of bats. And you can imagine the bats, especially the canopy-dwelling bats, they live in three-dimensional colonies, and so they're excreting urine and feces all of the time. So there's many opportunities for transmission amongst bats. How do other species get infected? There's a lot of work that needs to be done there because we never directly observe those events. So we put together a good, I guess, a parsimonious explanation given the data we have. For Hendra virus, we understand that horses are infected by grazing on pasture that's been contaminated by the excreta of bats that are feeding in trees within the horse paddock. For Nipah virus, we understand that humans get infected by drinking contaminated date palm sap that the bats have been consuming during the night, then they contaminate the pot that's collecting the sap. So for some pathogens, we really understand those pathways well, and then for other pathogens, we have no data whatsoever of what that pathway is.
Do we know anything about humans coming in contact with guano and how that may lead to transmission?
I haven't seen, we probably do, but I don't know. But I think that would be a very plausible pathway of transmission. There's also often these pathogens are transmitted through an intermediate host, and that intermediate host has more contact with the bat or with the bat excreta, and then builds up a high viral load and then has more contact with humans. I mean, horses and Hendra are a great example. Horses are like big vacuum cleaners, literally moving along the pasture, sniffing to make sure the pasture is not contaminated before they forage. They have a massive surface area of mucosa in the nasal passages, and then sniff up the virus. The virus travels probably up through the first cranial nerve into the brain and up some into the respiratory tract, and then that horse becomes a vesicle for human infection. So there's probably often these intermediate hosts involved.
And we heard at this meeting efforts to make guano collecting safer, right? Because it is a potential source of infection. Instead of saying you can't collect guano anymore, which is their livelihood, right? We say, all right, let's try and do it safely. So I think that's a nice approach. We also heard of bats taking a bite out of fruit and not liking it and dropping it, but contaminating it perhaps, and other animals picking it up. So,
or I liked the one where we heard about the bats taking the juice out of the fruit and leaving the pulp behind for someone else.
And someone else will eat it, right?
Right.
Yeah. these things you don't think about. All right, so there's a paper I want to ask you about, which you're both authors on. Novel Hendra virus variant in flying foxes in Australia. So Vincent, what's the significance of that?
I think the significance is twofold. One, obviously you always need to expect the unexpected. So preconceived notions of thinking what is circulating in your reservoir is apparently not good enough. And the strength of syndromic surveillance, like having a fail-back mechanism where, in this case, horses got infected and they detected a novel Hendra virus, which is, like, as Chris already mentioned, very similar to the first Hendra virus, but the primary probe was a mismatch, so we couldn't pick it up in our sample set. And obviously, none of the surveillance ever picked it up. So when that particular novel Hendra virus was identified, we actually went back to around 5,000 screened samples for Hendra virus 1 and rescreened all those samples then for Hendra virus 2. And then we actually saw quite some Hendra virus 2 positives as well. So I think, again, pointing out to the strength of like syndromic surveillance, picking up those diseases either in animals or in humans, combined with like a little bit broader sense of that you don't know really what's circulating unless you really start looking for it.
I'll add too, I think it's a good example of the efficiency of syndromic surveillance, because for the pathogen to be within that species, it's already gone through a whole series of filters from its reservoir host, that it's been able to overcome the structural barriers, innate immune barriers. It's been able to actually even cause disease within that host. And so we know that pathogen then is important, and then we can go back and backwards sample to the reservoir host and try to understand the ecology. And certainly there are efficiencies in that approach compared to going into the wildlife host and then trying to understand what the significance of the viruses that we newly discover are.
So roughly how many Hendra spillovers have there been, horses and people, over the years?
I think we're at 64 spillover events from bats to horses.
Okay, and your novel variant, Vincent, has that been involved in any of those?
Yes. Like there have been 2 detected spillover events of the novel Hendra, but it's also important to know that there are many horses every year that have a veterinarian called to them that have fever, respiratory distress, neurological disorder, and test negative for Hendra virus. And so we've probably been missing this novel variant for years.
So is Hendra virus only in Australia, or can you find it or something related to it elsewhere?
Yes, so very close relatives are found in Indonesia. And then there's a real spectrum of Nipah viruses across the old world tropics all the way through Africa.
And is that because of the bat hosts are similar?
Well, the bat hosts overlap. So we have Pteropus electo going from Australia, actually flying to Indonesia and back. Some nice tracking work actually demonstrated the bats crossing those international borders. And then the bats gradually change taxonomically across right through to the Western Africa.
But in the Americas, say, there's nothing related to Hendra, or we don't know?
Not in the North America, but there have been certainly paramyxoviruses and Nipah viruses discovered in South America. Very little known about them.
So you're both also authors on a paper, Nipah virus detection at bat roosts after spillover events in Bangladesh. So do the findings from that paper mean that the roosts were the spillover source? And what kind of implications does that work have on surveillance for viruses like Nipah or Hendra?
Yeah, I think I think it does point towards, I guess on one hand, the lack of success in trying to detect significant circulation of Nipah virus outside the moment where we actually detect spillover. So that is very significant, but obviously it's after the fact spillover takes place. So I think it makes still the system, from my end, a little bit hard to understand how you would actually really design true preemptive, other than obviously focusing at the date palm consumption and potentially pasteurization of date palm use, how to design preemptive tools to really stop that spillover.
So I understand that covering the date palm collection vessels is a good way to prevent spillover. So, but I understand also it's not done all the time. So why is that?
It needs constant communication, constant work. And so there needs to be major investment within the communities that drink date palm sap. And that's very difficult to maintain.
And I think it also points in how, as humans, how bad we are at like really appreciating preemptive techniques, right? So I'm at NIH, we really love our vaccines, we love our antivirals, but they're of course after the fact, after the pandemic, and I think there's still not a very good appreciation or even funding to understand how to like stop spillover in the first place. Or investment, for instance, from the World Bank, for instance. Obviously, if they're wet markets, there's a huge economy around that. So if you want to stop that, you probably have to give them a monetary incentive to stop that. So that, in my point of view, that should be very feasible, right? Given how much money these outbreaks have cost, like the $10 billion of the West African Ebola outbreak, the trillions of dollars Peter Daszak mentioned with SARS-CoV-2. So I think it's achievable. We just don't really appreciate it.
So Nipah seems to transmit quite a bit in pigs and less so in humans. Do we know why that is?
Oh, now they're looking at me. Yeah, I think like most of the pig work has, of course, like been a little bit anecdotal. So that's the outbreak in Malaysia. What I think you can actually see that probably is driven by a number of things. Obviously, the same as with humans, you have a very naive population. When the virus comes in, it does very well in those pigs. And of course, they're in closed quarters and very efficient in direct contact transmission. That might be partly similar in humans, but there might be subtle differences why ongoing human-to-human transmission, for instance, going from a chain from 1 to 2 to 3 to 4 to 5 is actually not as efficient in humans just because you don't have that contact network, or you have different mucosal surfaces. Again, like a pig respiratory tract is reasonably similar to humans, but of course their whole upper respiratory tract, the nasal turbinus, is completely vastly different, way bigger contact area. So there might all be these like subtle differences which facilitate easier transmission in the pigs versus humans.
Are there differences in the viral genetics in the viruses that transmit between pigs versus humans?
No, because I only think there's only one big outbreak. Correct me if I'm wrong.
That's right.
Well, we do have isolates, though. We have infectious viruses. And
yes, but then the people actually got infected via the pigs.
Right. So when that happens, are there sequence differences in the human versus the pig isolates that might be candidates for some changes?
So that is a very good question, but I think this happened in '98. So like now you would do that. So now you would have like all the next generation sequencing tools. You would set up your GridION or your Illumina platform, and you would actually specifically look at potential changes over time. At that time, there was like one isolate, one or two isolates, and that's what they sequenced. So you're kind of missing that information, what was happening on one hand in the pigs and in the humans. But yes, it would be very interesting, for instance, to see what's happening during a transmission chain, for instance, in either in Kerala, India, or in Bangladesh, to see are there any mutations which would pinpoint to host-specific adaptations. But I haven't seen that data yet.
And of course, since pig biosecurity has been improved, we're not going to see those kind of outbreaks any longer, most likely. Or at least we hope. In both of these papers, were you going to say something? I'm sorry.
No, but I think like what I pointed out, that's actually one of the bigger successes of like a very strategically preemptive strategy, right? You take away the intermediate host and you stop spillover.
Right, so the pigs were outside and now they're inside, and that makes a big difference, right? And you can still grow pigs, but you just do it in a safer way. So both of these papers, you mentioned, you write that this work could be important for identifying pandemic threats. So I wonder if you could speak a little bit about what are you looking for a pandemic threat? I don't know who's best to address that, Raina or Vincent.
I think we can go back and forth. I think Raina can do it from like a holistic population level, and I can look at it more from a virology perspective. So I think I think it would be good for you to start painting the bigger picture there.
Right. Well, it might only take one spillover for a pandemic, right? We have millions of cases, but that really is derived by perhaps a single spillover event. Certainly in Ebola 2014, the evidence suggests that a single spillover event led to over 28,000 cases, 11,000 deaths. So really, to be a pandemic threat, there has to be the opportunity for spillover, so there has to be the opportunity for exposure between that reservoir host and a competent recipient host. And then once you have that ability to transmit across species, then there's going to be a whole bunch of processes which I think Vincent can go into about what the virus has to be able to overcome to actually transmit within the new host.
Yeah, and I think like some of the data which was shown, particularly with Cedar virus in combination with like Hendra virus and Nipah virus, where Cedar is like a non-pathogenic Nipah virus, I think that really points towards some of the intrinsic details what makes a virus successful and what doesn't make a virus successful. In the case of like Nipah viruses, they all bind to efferents, that's a receptor. It's very conserved across species. So for a virus, that's a very good strategy. It's very similar to ACE2, for instance, where there's also quite some conservation. So on a receptor level, that virus can use all these different receptors in these different species, but that's just the starting point, right? Then you have to get in, and then you have to interact with the host cellular defense. And I think that's the big step. And if you cannot efficiently block, or during those initial phase of replication, mutate to effectively block that innate response, you're not going to be successful. You're just going to be cleared by the host immune response. So I think that's a little bit the next frontier in trying to understand the diversity of viruses, but how can you truly weed out what is going to be like only initial spillover, but what actually really captures that ability to cause disease, which is the first step. So we're still talking about spillover, but more importantly, human-to-human transmission. In the end, it's all about human-to-human transmission. We're like, as global health, you're not that really interested in that one single case. You're interested in when that thing takes off.
So basically what we can do right now is we have an isolate. We can see if it will infect human cells in culture. And that's it. All the other things you said are very important, but we really don't have a lot of ability. We could look at antagonism of interferon, for example, but we know nothing about transmission, right?
No, that's absolutely correct.
Yeah.
So given the potential of these viruses in causing pandemics, should we be working on vaccines and therapeutics?
Well, I think we're already working that. Like most of the coronavirus therapeutic actually have quite a broad ability to interact with a wide variety of viruses. So I think we're already there. Obviously, what we did need was a pandemic to move these potential antivirals through clinical trials. So that's the big step, and you need to have significant circulation before you can actually move that through. And I think that's now similar with monkeypox, where there are at least a couple of antivirals, but not I think like at least TPOX have not been completely through phase 3 clinical trials. So NIAID is now setting up these clinical trials to really move them through. So that's one. So you would have like broadly active antivirals, which I think there's a lot of investment now. And then of course there's like talk about like pan-coronavirus vaccines, or and we've been working on that like in the past with avian influenza, really get like a diversity of hemagglutinins ready to move into, that was classic, like the classic egg-grown vaccine, to be able to move that quite rapidly. So I think everything is kind of in place, and I think the ideas are there. There's going to be a lot of investment in what we call like prototype research. So for instance, we want to invest in a rhinovirus vaccine. So you take Lassa virus as your prototypic virus to actually do antivirals and vaccines. With Nipah virus, you take Nipah or Hendra, and so on. So there's a lot of investment going on in that, but then fact of the matter is, like, you still need to move them through clinical trials.
Sure.
Where you need cases, right?
Yes.
Enough cases to give you
Yeah, although there's
result.
of course like the system with currently, which is a long trajectory, which is like the animal rule. So the current Guinea's monkeypox vaccine, that actually went through an animal rule approval system. So we have the ability to move things through, but it's a very long process.
What about focusing on vaccinating or therapeutics for the intermediate host?
But then you need to know the intermediate host. Right? Like sometimes that's relatively easy. So for instance, of course, the horse vaccine strategy in Australia is a very good example. We have been talking about, for instance, vaccinating dromedary camels in the Middle East. If you then actually look at the population structure, and I'll show some data today in my talk, but they have like very rapid waning immunity, so your vaccination might not be particularly successful. You might need to target a very specific part of your intermediate host, for instance, the young dromedary camels, which you probably only can truly start vaccinating after 6 months of age. So in theory, it's a very strong principle. Practically, there also needs to be like an economic incentive, right? Like you need to convince the camel herder that he needs to vaccinate his animal against something the animal doesn't really get sick from.
All right, so one more bat question, then we'll move on to spillover and ecology. So do we understand how bats are infected? So in humans, we look at acute and persistent infection. So are bats acutely infected, or do they shed their lifetime? Are they reinfected multiple times? Raina, what do we know about this?
It's one of my favorite questions.
Good.
We really do not know. We have all sorts of pieces of evidence that I think the most parsimonious explanation for what we see with the dynamics of Hendra virus in the wild is that bats are persistently infected, or at least some bats are persistently infected. Perhaps we have a combination of persistent infections and then some what we call SIRS dynamics, where animals lose their resistance over time and can be reinfected. I often wonder if Hendra is something like a hepatitis C kind of virus. Like some bats are able to clear it, and some bats just can't quite clear it, and then they are infected for some period of time. We really don't have any direct evidence that is the case, though. It's very hard to do those kinds of experiments where you hold animals in BSL-4 for a long period of time and reinfect and so on. One thing we do see in the field, which is really interesting, this is work by Alison Peel in Australia, is that when the bats are shedding a lot of Hendra virus, they're also shedding many other types of viruses. For example, Alison showed the bats were shedding 9 other paramyxoviruses at the same time as a big pulse of Hendra. And that suggests that maybe there's some sort of a common environmental driver of all of these viruses. So stress and perhaps reactivation of latent infections may be driving the shedding of the virus, and perhaps then be a part of the spillover process.
And some of the drivers of these pulses that were talked about at this meeting, nutrient availability and hormonal levels having to do with reproduction?
Exactly. So with Hendra virus, we see this very clear seasonal pattern where it's actually hard to find the virus most of the year. And then in winter, we see the bats shedding the virus. But we see them shedding more virus if that winter follows a food shortage, so the bats have been nutritionally stressed. And we see them shedding more virus if they're in a habitat that they wouldn't usually occupy in winter. So habitats that we think are suboptimal for bats. So again, it's just this like epidemiological evidence that there's perhaps some kind of a stress involved in shedding of these viruses.
So I found this part of your talk particularly fascinating. So can you talk a little bit about types of human activity or land use or things like that might influence shedding or some of these pulses in bats? Are there things that we're doing that might make pulses or spillovers more likely?
Yes, there's a lot we're doing that might make spillovers more likely. If you think about what has to happen for a spillover, you have to have a reservoir host. It needs to be infected, and then it needs to be shedding the virus, but in proximity to another host. often, as we discussed, an intermediate host, but it could also be a human. And so many of the things that we do make those processes more likely. So we remove habitat, we change the climate, we put these animals under more and more stress, we may make them energetically stressed as well as nutritionally stressed. And physiologically stressed, and we know that that can increase the amount of the likelihood of shedding virus and the likelihood of shedding a lot of virus, and it also changes the behavior of animals so that they're more likely to be in proximity with humans. So seeking out new food sources in human environments, or even humans actually even encroaching on their environments and coming into contact.
I liked your data also about changes in the frequency of flowering and winter flowering and how that played a role in this. So can you talk a little bit about that and how human effects may play a role on that?
The Hendra virus system is very interesting in that we've discovered that the flowering of the habitat that flowers in winter and produces nectar is very important for the ecology of the bats, and it may be really the big bottleneck in their whole life cycle. That having enough food in winter is perhaps maybe the trigger for bats to move into agricultural and urban areas to seek more food. But when we do see flowering of these habitats, of remnant patches of habitat, we do see a lack of spillover events. That work is not yet published, but it will be coming out soon.
Cool.
But I do think it's really like a prime example how we need to invest in long-term multidisciplinary studies, because the amount of people you get around in a research team to actually address these research questions, I think that's a phenomenal achievement. And it really, like for the funders here in the room, I know there's some NIAID program officers, like we need to have more funding for these kind of studies which go 10 years, 15 years, 5 years. Like a normal grant cycle is not good enough to do these kind of research.
Yes, it took 25 years of data to see those broad patterns within the Hendra system. And there are very few systems with that level of data.
So Raina, in a commentary you just wrote, you said there are 4 actions we need to do to reduce spillover from some of these reasons that you just spoke of. Tell us what those are.
So one of those actions is really what our group is focused on, and that is trying to preserve and restore the habitat of the reservoir hosts of these viruses. And for just the reasons that I mentioned, that if the animals have complex adaptive networks of landscapes, they can find the food that they need so that they don't suffer nutritional stress or physiological stress. They don't have to move into human-dominated areas to find resources. So all of these processes that increase the probability of pandemics will be reduced. Unfortunately, those kinds of measures are really not on the table at the moment as pandemic prevention. Like our focus is on early detection, vaccination, and therapeutics, which is, we must have therapeutics and vaccination, early detection. They're all very, very important measures. But the actual prevention of the spillover has almost been deemed too difficult. In fact, there was an advisory group to WHO who recently said that we should not invest in pandemic prevention because it was, to quote, like boiling the ocean. And I think that is really a shame to hear that, because if we can get upstream of these spillover events, that is the most efficient way to stop the next pandemic. And again, think Ebola 2014, all of those cases, one spillover event. Maybe this pandemic derived from a single spillover event from a bat to another species. If we stop that one event, we stop the entire process. But once the pathogen is spreading within the human population, it's really difficult to get on top of it. As we've seen, it takes a while for the vaccines and therapeutics to go through trial, and by the time that you have effective interventions, the cat's out of the bag. So there does need to be a real focus on this. I think the way that we need to do it is we've really got to understand the mechanisms of spillover. There's been very little work on how this spillover event happens. But once we start to understand these complex ecological mechanisms, we can start to see efficient ways of intervening for example, in the Australian system, we're not saying stop deforestation, we're saying plant 5 species of eucalypt. If we plant these 5 species, they flower in winter, they provide excellent nutrition for bats, and we have really good evidence that if we replant these 5 species, we could actually stop Hendra virus spillovers. If we had that kind of information for coronaviruses, for Nipah virus, for many of the undiscovered viruses that we're looking at right now, maybe we could stop spillover events very efficiently.
So does the biodiversity in a particular area influence spillover potential?
Well, in infectious disease ecology, we're always talking about the dilution effect. And as a reminder, that is a theory that a number of groups, including Rick Osveld, Felicia Kiesling, have talked about for many years, where a vector-borne pathogen, if you have a lot of biodiversity, many hosts that are not very competent for the pathogen will dilute out the bites of the vector. But if you have a system without much diversity, you often have these highly competent, competitive, dominant hosts that take all the bites of the vector, and therefore you see an increase in the pathogen prevalence. But I think biodiversity is important in a much more profound level than the few vector-borne diseases in which it's been studied for. We should think about diversity as really as whole, complex ecosystems. And if we had systems that were resilient and diverse, then we'd have places for animals to go, places for animals to move, nutrition for animals at all times of year. If we had a climate extreme, animals would be able to find something else to eat. If we had some sort of event that caused animals to move, like climate change, there'll be places for them to go. So we need to be thinking of diversity as these complex adaptive systems. And so we need to be really preserving large landscapes, large connected landscapes to keep reservoir hosts healthy, to keep us healthy.
Yeah, and I think in that sense to think a little bit more, like as a virologist, a little bit more simplistic, I guess. Like if you have a reduction in biodiversity, that has causes, right? That could be deforestation, road building, which would directly increase spatial connectivity. So it's not necessarily the spillover per se, but the chance of the spillover reaching, let's say, Kinshasa 6 hours to Paris, another 8 hours to DC, right? So I think you can like more rudimentary think about a reduction in biodiversity suggests something is like going on there, which might actually facilitate spillover more.
I hope my students who'd complain about having to learn some parts of biology other than molecular listen to that.
So let's take a simplistic view from another virologist, right? So we heard a talk where there are bats living at a particular roost site, and at night they go to a particular tree to get some food. And it just so happens that people have built houses next to that tree. So what could you do about that? Is there a solution?
I thought their data was just amazing. And the fact that you could show someone that this bat moved to your tree in your backyard and was infected with Marburg virus, I think, and ate the fruit on your tree. I think that's extraordinary. And maybe it's that sort of communication, that sort of risk communication would be important. Of course, that could also backfire, and you could see bats being persecuted. And I know the CDC group that presented that work also said that they had bats persecuted that were infected with Marburg, and they saw the infection prevalence with Marburg double after that group of bats was culled. So we have to be very cautious in how we present these situations.
It's just one tree and a few houses, and so it's a difficult problem on a large scale. You can't plant trees elsewhere to divert the bats. You couldn't possibly do that everywhere, right?
Well, we possibly could. Planting trees is actually, it's very cheap, it's easy, it has a lot of benefits for biodiversity and climate mitigation. The key is that you need to understand the ecology of the system to know whether it would work. In Australia, we know that planting trees that flower in winter would work because we've done the deep ecological studies looking at the movement and the preferences of animals, and we see when there is a big blossoming event that the animals fly over the agricultural areas, they fly over all the weedy species they're usually feeding on to go to these native forests to feed on nectar. But those kinds of studies would need to be done all around the world to know what drives animals away from humans. What are their ecological needs that would allow them to subsist without having to come into our environments? So we need a lot more, we need investment in very basic ecological research, basic natural history, to be able to know what are the key interventions that need to be made.
In fact, in one of your commentaries, you make the point that if you plant the wrong tree, you attract the wrong predators. So you can't just plant any trees in a city, for example.
Right, yes. Well, you wouldn't want to plant the right trees in a horse paddock. That might not be a good idea.
That's right. Exactly right. It's your turn, Brianne.
It is my turn. So I'm jumping to one of our sort of questions near the end, just because you guys are making me think a lot about the things I tell my students and the things I want to tell my students. So we have many listeners here who are virologists or who are new to a lot of the information that you are talking about today. So what is one message that you would really like all of them to have?
I think, like, if you study virologists, think holistically. It's beautiful. It's like, I love it a lot. Like, being able to work with people like Raina and a lot of collaborators here in the room really expanded my vision. And really, like, starting from a molecular road, sometimes our understanding is very simplistic. And I think if you then delve into these, like, ecosystem changes and link that with the virology, I think it's a very strong concept. And it's just an amazing lot of fun.
Well, you know, before you answer, Raina, if you're trained in molecular biology, you don't think that way. You think of what's in that little tube. You don't even think of what's outside the door, right? And that's what we're talking about at this meeting. That virus in there, how it interacts with its natural host and with everything around it. And that can only happen when people who study the outside mix with virologists or whatever the pathogen is. And that's why a meeting like this is so good, right?
It's like there's a reason why all parts of biology are important.
But typically that's how I build my lab as well. I have people coming through who are very molecular oriented. I always take them into the field. They don't necessarily have to have a complete field project, but they need to see what's happening to appreciate that, and then they can design better experiments and better research questions.
That's awesome.
Okay, Raina.
I have to build on that because that's a really great comment by Vincent. It reminds me actually of one of my first Hendra virus meetings. It was after a big cluster of Hendra virus spillover events, and we assembled a bunch of experts together who worked on the bats, the virus, the horses, and we talked about what drives spillover. And I came away really shocked because everyone thought they had the answer of what was most important. The virologists, absolutely, it's the virus, it's the survival of the virus, that's what was important. The bat ecologist, no, it's a change in the behavior of the bats. The horse people, no, it's the way that horses are managed. And then a guy turned up in overalls from his horse property. He was an agronomist. He grew grass for horses, and he said, no, it's the grass. The grass is absolutely most important. And he had a really good theory for that. And I came away from that meeting and I realized that actually they're all right. Every one of those processes is really important, and what was critical was to understand how those processes all align to either facilitate or stop spillover. And I think we really came out with very deep insights from that meeting, and that's inspired me to keep doing this transdisciplinary work, just look at these systems in all these different perspectives, and it's led to some really important insights.
So I want to go back to something in one of your papers that I think is fascinating. You say that there's a propensity for small-bodied animals to predominate in human-modified environments. Can you explain that?
Right, that is not my paper.
But you could have written it.
I think that might be a paper by maybe Gibbs recently. I think it came out during the pandemic, which is a really, really nice paper showing that hosts that are more likely to be competent for zoonotic pathogens are also more likely to live in urban environments. And I cite that paper all of the time. Yes, and I think the theory there is that small-bodied hosts are investing everything in reproduction, right? Because these are selected species, and they're not investing energy in immunity, and therefore they're able to host these pathogens and maybe shed high amounts of pathogen. I think that's the theory. But it seems to play out at an international level and be a really important process.
Does that play into things like habitat fragmentation and a smaller animal being able to be in a fragmented habitat?
Yes, very much so. It does, because as we modify habitats, we remove predators, we remove all of the complex interactions amongst animals. We tend to see some species predominating, and they are these competitive dominant species, often rodents. What are they here? They're pigeons and rats, but they often are animals that also seem to be good at hosting zoonotic diseases.
I think it's also interesting because that's one of kind of like these fields where I think like laboratory and medical science and ecology kind of like bunch into each other. Because if you look at like a lot of like all the mouse data and the vaccine data, it does suggest that like most of the rodents are actually perfectly capable of mounting a very productive immune response. So I think also like people who write these people go back to the medical literature, to mouse studies, and trying to understand a little bit more on the mechanistic of the immune response and challenge studies, because I think there's so much to learn from both ends. Because I think there, like some of these concepts are clashing a little bit, and they can form testable hypotheses. So if you pose a hypothesis like that, I think that's perfectly testable. So then get to the next step and actually test that hypothesis.
Should we get some colonies of some larger bodied animals so that we can make the comparison?
For instance.
Would this explain why small mammals predominated after the dinosaur extinction?
You are way outside my expertise there.
That's good. You should stay in your lane.
I'm old, but not that old.
It sounds good, right? Apparently. But, all right, so I'm getting from this idea that we need to reduce our fragmentation, our encroachment. It's hard because the human population is growing. People need to eat, they need to work, so there's got to be some balance, right? That's your message. You don't have to cut out all the land repurposing, but you can reduce it and do some other things. But can we also learn to live with our non-human neighbors in some way? So Vincent, you said before that some bats are peri-domestic, right? They're perfectly happy living near people, right? Near their homes.
Yeah, but I think like if you look, for instance, at Europe, like if you have like a bat roost in your house, you cannot move them. So I think like in a lot of instances, people are very comfortable with bats having around. So it's kind of like trying to understand where and how they would be a problem. Like mostly probably for 90% of the species, it might actually not be a problem. But you might not want to have, as Raina already mentioned, like a big tree in a paddock full of horses and really attracting like Pteropus bats.
Okay.
Well, even with Pteropus, we have only seen the spillover of Hendra virus really since 1994, but we've seen climate extremes and food shortages in bats for probably forever. But it wasn't until we removed enough winter habitat and we saw bats moving into agricultural areas, we see spillover. And I suspect that in many parts of the world, people have lived with bats forever and there hasn't been a problem. But as we then perturb those systems, we drive animals into stressful situations in proximity to humans, we build roads through, like we're doubling the perimeter of roads in the next 25 years as a projection. So we're building roads through all of these habitats. We're bringing new populations in that haven't been exposed to wild animals before. We're just changing things so rapidly that we're seeing things that we would never have seen 20, 30 years ago.
So Raina, would you say that this is related more to kind of changing the frequency of the pulses or the stressors on the animals, or can we live near them if there weren't so many pulses and stressors on them?
We're both changing the amount of virus, the likelihood of shedding virus, the amount of virus animals are shedding because we're putting them in stressful situations, but we're also making that happen in a situation in proximity to people. So if you think, like, if a bat pees in a forest, does anyone care? If a bat pees into your date palm sap, then everyone cares.
Oh my gosh.
New philosophical question.
We may have a title.
I think we might.
I want to just paraphrase again Peter Daszak, who said, you can't just write strong letters to implement these changes. So can we do something more than that? What has to be done? Because it's a big ask, right?
It's inconceivable to me that we're not doing so much more given the impact of this pandemic, and trying to at least understand how these events happen so that we can efficiently address them, but address them right at that source of the spillover event. It's hard to understand why we're not investing huge amounts of money. How we make that happen, I don't know. That Peter Daszak is really good at figuring that out. We need really good advocates as speaking to politicians that are talking to the groups that are making these international treaties right now on pandemics. We need really, really good communication.
But I also think we have already 3 very cautionary tales. If we don't do it correctly, obviously we had a pandemic. The widespread of H5N1 now in wild bird populations across the world. I did my PhD in avian influenza, so did a couple of infection experiments with chickens. It's a really nasty virus in chickens. A couple of weeks ago, I got a text message from my staff scientist, Niltje, from her dad, who was showing very proudly a dead chicken in his backyard. And I said, do not touch that chicken. That chicken died of H5N1. Somebody, just in the backyard, in the Netherlands, H5N1. And it's now everywhere. Cautionary tale. If we don't do this, this is what's going to happen. Third, monkeypox. We've known it for a while. We've had vaccines. We have advanced. Nobody bothered to bring that into Africa. None. And see where we're at. Right? Again, 3 tales in a very short time. It's actually quite astonishing how unable we are to deal with these bigger questions.
I think part of the solution is getting more people to know that, because right now that is not the way it's presented in the mass media, for example, and so that's why what we do is, I think, more important. But I would like to raise the positivity level here at the end. And we're going to ask you 4 rapid-fire questions, and we'll let Brianne start.
All right. What is your advice for young scientists?
Be very curious and follow your dreams, follow what you're interested in and passionate about.
Look for the right mentors. There are tons of them, and typically they're awesome. And scientists love to talk about their science.
If you could have dinner with anyone, living or dead, who would it be?
I choose Raina.
Yeah, Vincent, I'll have dinner with you.
This is too easy, but okay, we'll take it.
All right. What would you have done if you weren't a scientist?
A weird story. I was a gravedigger for a while. But I would probably be either a plumber or a bike mechanic.
I'm not sure we've ever had gravedigger for that one.
I would be a documentary filmmaker. I'd travel around the world, maybe doing David Attenborough films on the camera side, just to be in beautiful places.
And finally, what keeps you up at night?
Well, I think you wanted us to be positive, I thought.
The first 3 were.
Well, it's this combination of climate change, loss of biodiversity, and pandemics they're all wound in. It's 3 converging crises. They're all really driven by very similar phenomena, and it keeps me up at night, certainly wondering how we can address these really seriously. They threaten our existence.
Yeah, I typically sleep quite well, but there were these instances, particularly during the pandemic, where everybody felt the need to provide not particularly constructive criticism to the work we're doing, whether we were doing it on disinfection, stability, whether it was vaccines. Everybody just needed to pee over the work and not particularly useful. And I think that, like, dude, step back, relax, read the paper. I'm perfectly fine in discussing it, but I'm not going to do it on Twitter. I'm not on Twitter. So that was, for me, like, very unhelpful.
Okay. That's TWiV 923. You can find the show notes at microbe.tv/TWiV. If you have a question or a comment, TWiV at microbe.tv. If you enjoy our work, we are a nonprofit company, and we exist on your donations. You go to microbe.tv/contribute. Vincent Munster is at the NIH Rocky Mountain Laboratories. Thank you, Vincent, for coming back.
More than happy to. And if anybody wants a postdoctoral position, feel free to email me. We have positions opening in a lot of different areas, transmission, fieldwork in Africa, so just shoot me an email.
Raina Plowright is at Cornell University. Thanks so much.
Thank you.
Brianne Barker's at Drew University, BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here. Thank you both. I learned a lot.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank Tony Schounts for having TWiV at this meeting. Thank you, Tony, and the meeting organizers for agreeing with him. And I'd also like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Jolene for the timestamps, and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. Thank you.